Bacteria, some of the oldest life forms on our planet, are engaged in a constant silent war with viruses known as bacteriophages. This battle has driven the evolution of sophisticated defense mechanisms within bacteria. At the heart of one such system lies CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. These peculiar DNA sequences were first observed in the late 1980s, but their function remained a mystery for many years. Scientists discovered that these spacers are segments of DNA captured from invading viruses, forming a genetic library of past infections. Adjacent to these CRISPR arrays are CRISPR-associated genes, or genes which encode proteins that enable the CRISPR system to function. These Cas proteins perform various roles, from acquiring new spacer sequences to neutralizing threats. The discovery of the CRISPR-Cas system represents a fascinating journey into microbial defense, now ingeniously adapted by scientists to edit genomes with unprecedented precision. This ancient bacterial immune system has opened new frontiers in medicine, agriculture, and basic research. The first crucial step in the CRISPR-Cas immune response is the adaptation phase, where the bacterium acquires a memory of a new viral invader. Specific Cas proteins, notably Cas9 and Cas2, identify the invading viral DNA and excise a small fragment from it. This fragment becomes a new spacer integrated into the CRISPR array within the bacterium's genome. The new spacer is inserted near a special sequence called the leader sequence, pushing older spacers further down the array. This orderly insertion creates a genetic memory of the specific virus. Should the same type of virus attempt to infect the bacterium in the future, this stored spacer will serve as a template for recognition. The bacterial cell learns from its encounters and prepares for future threats, passing this immunological memory down through generations. By continuously updating its CRISPR array with new spacers, the bacterial population maintains a dynamic and evolving defense portfolio. The CRISPR array becomes a living history book of past infections, equipping the bacteria to identify and neutralize threats swiftly. Once the genetic memory of a viral invader is stored as a spacer within the CRISPR array, the bacterium must access and use this information when a threat reappears. The CRISPR array is transcribed into a long RNA molecule known as pre-CRISPR RNA, pre-cRNA. Specific Cas proteins or other cellular enzymes cleave the pre-cRNA into mature CRISPR RNAs, cRNAs. Each mature cRNA contains a single spacer sequence flanked by portions of the repeat sequence. These key RNAs guide the defense machinery to the target viral DNA. In type 2 systems, another RNA molecule called trace rRNA binds to the repeat regions of the pre-cRNA, forming an RNA duplex. The mature cRNA, often in complex with trace rRNA, is now primed to seek out its target, ensuring the specificity of the immune response. With the guide RNAs prepared, the CRISPR-Cas system enters the interference phase, where the bacterial cell actively defends itself against the invading virus. The mature cRNA, often complexed with trace rRNA, associates with Cas9, an endonuclease capable of cutting DNA. The guide RNA directs Cas9 to its designated target sequence within the viral genome. The Cas9 guide RNA complex patrols the bacterial cell, searching for DNA sequences that match the spacer region of the guide RNA. If a complementary sequence is found, the complex locks on and Cas9 cleaves the DNA, neutralizing the virus. The requirement for a PAM sequence ensures that Cas9 only targets foreign DNA. This precise molecular scalpel, evolved by bacteria to combat viruses, is now repurposed for gene editing. Scientists harness its ability to make targeted cuts in DNA at virtually any desired location. The realization that the bacterial CRISPR-Cas9 system could be programmed to target specific DNA sequences was a watershed moment in molecular biology. Scientists quickly understood that this system could be redirected to find and cut any DNA sequence in other organisms by changing the guide RNA sequence. This transformed a bacterial defense mechanism into a versatile and powerful genome editing tool. Researchers designed a synthetic single-guide RNA, sgRNA, combining the essential features of both the cRNA and trace rRNA. To target a specific gene in a eukaryotic cell, scientists introduced the Cas9 protein and the custom-designed sgRNA. When Cas9 makes a double-strand break in the DNA, the cell's natural DNA repair mechanisms are activated. One repair pathway, non-homologous end joining, NHEJ, often introduces small insertions or deletions, disrupting the gene's function. Alternatively, homology-directed repair, HD,